Why would two nations sharing the world's longest undefended border, speaking the same language, and descending from many of the same European ancestors be genetically different? The answer, hidden in American and Canadian DNA, reveals a story of selection, migration, and historical accident that scientists are only beginning to understand. In 2009, researchers at the University of Toronto published a study that quietly shocked the genetic community. After analyzing DNA samples from thousands of Americans and Canadians of similar European descent, they expected to find populations virtually identical at the genetic level. Instead, they discovered measurable differences in allele frequencies, disease susceptibilities, and even behavioral genetics between the two populations. How could people separated by an invisible line develop genetic distinctions in just a few centuries? The answer requires understanding that genetic differences don't require thousands of years to emerge. They require selection pressure, founder effects, and differential migration patterns. America and Canada experienced all three in dramatically different ways, creating populations that look similar on the surface but carry distinct genetic signatures underneath. Before understanding modern American and Canadian genetics, we must acknowledge the Indigenous Foundation upon which both nations were built. Canadian Indigenous populations, particularly in the far north, show higher frequencies of genetic variants adapted to extreme cold. The Inuit carry unique variants in genes like CPT1A, affecting fatty acid metabolism, that allow efficient energy extraction from high-fat diets. These variants appear in approximately 68% of Inuit populations, but are virtually absent in most American Indigenous groups further south. The genetic diversity of Indigenous peoples in the continental United States was shaped by different environmental pressures. Southwestern tribes show adaptations to heat and drought. These Indigenous genetic foundations form a baseline difference between the two countries that persists today through admixture in mixed ancestry populations. The most significant source of American-Canadian genetic differences lies in who founded each country. The United States received its primary European founding population from England, but also drew heavily from Germany, the Netherlands, and Scotland from the 17th century onwards. These populations came from specific regions of Britain and Europe, carrying the genetic variants common in those specific places. Canada's founding European population was strikingly different. French colonists from specific regions of northwestern France, particularly Normandy, Brittany, and Poitou, settled Quebec beginning in the early 17th century. Approximately 8,000 French settlers who arrived before 1760 became the ancestors of over 7 million modern French Canadians. Dr. Damien Labouda at the University of Montreal spent decades studying this French-Canadian founder effect. His research revealed that modern French Canadians carry genetic variants at frequencies dramatically different from both modern French people and Americans. Certain rare genetic mutations that occurred in the original founding population became common simply through random inheritance in a small, isolated population. The French-Canadian population represents one of the most powerful natural genetic experiments in human history. After the British conquest of New France in 1760, French-Canadian communities became largely isolated, practicing endogamy and maintaining extraordinarily high birth rates. This combination of founder effect and subsequent isolation created what geneticists call a genetic bottleneck. Hereditary tyrosinemia type 1, a liver disease, occurs at rates of 1 in 1,800 in Quebec's Saguenay-Lac-Saint-Jean region, compared to 1 in 100,000 globally. Familial hypercholesterolemia appears at 4 times the global average in French Canadians. Americans, drawn from more diverse founding populations and experiencing continuous immigration throughout their history, never developed such concentrated genetic peculiarities. One of the most fascinating chapters in American-Canadian genetic divergence involves the American Revolution. When the 13 colonies declared independence in 1776, approximately 60,000 to 80,000 Loyalists fled north to Canada, settling in Nova Scotia, New Brunswick, and Ontario. This wasn't a random sample of the American population. Loyalists tended to be wealthier, more urban, better educated, and more Anglican than the revolutionary population that remained. Dr. Sherry Farrell-Rissett found that this Loyalist migration created a distinct genetic signature in maritime Canada, bringing specific British genetic variants underrepresented among rural American settlers. The revolutionary population that remained in America underwent its own selection. Those who stayed were disproportionately younger, more rural, and more willing to take risks than the Loyalist emigrants. Perhaps the most profound source of American-Canadian genetic difference is the legacy of slavery. Between 1619 and 1865, approximately 400,000 enslaved Africans were brought to the American colonies. Including natural population growth, enslaved people numbered nearly 4 million by the Civil War. Modern African Americans carry approximately 75 to 80% West African ancestry, combined with 20 to 25% European ancestry. African Americans today represent approximately 13% of the American population and carry distinct genetic markers, including elevated frequencies of sickle cell trait and certain cardiovascular variants that are rare in comparable Canadian populations. Canada had slavery too, but fewer than 4,000 enslaved people lived in British North America at slavery's peak. The genetic impact on Canada's overall profile was correspondingly smaller. America's great immigration waves of 1880 to 1920 brought primarily Southern and Eastern Europeans. Italians, Poles, Russians, Jews, and Greeks flooded through Ellis Island, bringing Mediterranean and Eastern European genetic variants into the American gene pool at frequencies that never occurred in Canada. Canada's immigration policy during the same period actively discouraged Southern and Eastern European immigration, preferring British, Scandinavian, and Northern European settlers. The consequence is measurable. Americans show significantly higher frequencies of genetic variants common in Southern Europe and the Middle East than Canadians do. One of the most significant sources of American-Canadian genetic difference is the Hispanic population, representing approximately 18% of Americans, but only about 5% of Canadians. Hispanic Americans carry a unique genetic mixture of Indigenous American, Spanish European, and West African ancestry. Studies show Hispanic Americans carry approximately 40 to 60% Indigenous American ancestry, 30 to 45% European ancestry, and 5 to 15% African ancestry. This large population fundamentally affects American genetic statistics, with variants common in Indigenous American populations appearing at higher frequencies in Americans than Canadians. Perhaps the most controversial aspect involves behavioral genetics. Americans are statistically more individualistic and risk-tolerant than Canadians, according to decades of cross-national psychological research. Some behavioral geneticists suggest this reflects mild genetic selection. Migration itself selects for certain traits. People who migrate tend to be more risk-tolerant and novelty-seeking than those who stay. The 7-repeat variant of the dopamine receptor gene, DRD4, associated with novelty-seeking and risk tolerance, shows slightly higher frequencies in populations with longer migration histories. Some studies have found marginally higher frequencies in American populations compared to Canadian ones, though the research remains contested. The genetic differences between Americans and Canadians manifest most clearly in disease statistics. Hereditary hemochromatosis shows higher frequencies in Americans of Northern European descent than in Canadians, possibly reflecting different proportions of Irish and Scandinavian ancestry. The HFE gene mutations causing this condition appear at slightly different frequencies even in populations of comparable ancestry. Pharmacogenomics has identified that Americans and Canadians metabolize certain medications at different average rates, reflecting the distinct ethnic compositions of the two populations, with real implications for medical treatment standards. Americans and Canadians are genetically different because history made them different. Different founding populations, different immigration patterns, different forced migrations, and different political events all created measurably distinct genetic profiles in two nations that share a border, a language, and much of their history. The differences are not dramatic. Americans and Canadians remain far more genetically similar to each other than either is to populations in Asia or Africa. But the differences are real, measurable, and consequential for medicine and public health. The story of American and Canadian genetics is ultimately a story about how history writes itself into our bodies. Every migration, every political upheaval, every cultural choice about who to welcome and who to exclude left genetic traces that persist in modern populations. Understanding these genetic differences reveals the fascinating complexity of how two closely related peoples developed their own distinct biological identities in just a few hundred years of separate history.